Ebola and a bit of the history and epidemiology, as well as details on the biology of the virus itself. And so this first table shows you a chronology of the largest Ebola outbreaks since 1976, when the virus appears to have first made the jump from an animal to the human population. And so in 1976, there was a big outbreak in Zaire, which is now referred to as the Democratic Republic of the Congo. The reported caseload was pretty low, but the death rate was extremely high for that low caseload, giving a death rate of 88%, which is particularly scary, especially since the seasonal flu has a death rate of about 0.1%, and SARS-CoV-2, which we're all freaking out about right now, has a death rate of about 0.2 or 0.3 overall. And so in 1976, there was another outbreak with a slightly what appears to be lower virulence version of the virus, but you never know. When your data sets are so low, it's hard to say whether these numbers are real, because you could get it could be based on luck, in other words, or chance. And so your numbers obviously could change significantly as more cases and deaths are reported, but fortunately this one sort of burned out quickly. Then again, the DRC was hit, and so you see the death rate go down and then back up in this one, and then down and then back up in this new case, and then level off somewhat in this particular case, and then in this particular outbreak. And then in 2014, there was a very significant outbreak in West Africa. In this particular case, at the time of this particular table being produced, there was 8,000 cases and nearly 4,000 deaths, and so that gave you a death rate of about half. But and I put this information here just so you can get a sense for what was being reported. Eventually, as the outbreak ended, there was about 30,000 reported cases with about 11,000 fatalities, for about a 40% death rate. The 2014 outbreak, as I mentioned, was centered around these West African countries: Liberia, Sierra Leone, and Guinea. And these particular nations were primarily where the virus hung out. We didn't get a lot of spread, which was surprising, because they actually, kind of shut the borders and made sure that there wasn't a lot of spread. There was a case where a few healthcare providers from Western countries like the United States had come to help out, and then tested positive and went back to the United States and were immediately quarantined and treated, but that set off a massive panic wave of individuals and news headlines about what if the Ebola goes airborne and it'll kill millions of people, and everybody freaked out for a little while. But then people stopped paying attention, and the news stopped reporting it or reporting these kinds of headlines, which is kind of what we see now with COVID-19. Everything's sensational and overblown as far as I'm concerned. So if we look at a bit of the epidemiology, the actual and predicted cases, over here on the y-axis is total number of cases, and then these figures here are total reported cases. And so and then eventually where the data stop, they have to make predictions using these fluctuation cones. And so again, in this particular case, you see that the fluctuation cone suggests that at the end, that at least for the foreseeable, from September to the next month, you can expect maybe 15,000 to 39,000 cases. Here's the fluctuation cone. And so the data are typically based on mathematical models, and again, mathematical models are only as good as the input data. And so in this particular case, when we a few years later, looking back, there were actually 15,000 lab confirmed cases with a total of 11,000 deaths. And so again, you can do the math on that, and at the time they were reporting 28,600 cases, but this is the lab confirmed. So one of the big questions was, what is the major reservoir for Ebola? And one of the main culprits was suspected to be bats. Bats tend to, just like with SARS-CoV-2, bats tend to be reservoirs for lots of different kinds of viruses. They have a very vigorous immune response, and so they're able to keep these deadly viruses in check. And they have a sort of a mutual, not a mutualistic, but a in a sense, maybe even a commensal relationship with these guys, with these viruses. And so some of these bats are butchered and eaten as bushmeat, and it's not surprising then how the bats' viruses can get into hosts. And so this is a pretty big fruit bat. And so you can see some of these open air markets, these that produce preparing bushmeat. Here's a whole bunch of different kinds of rodents, and you can see these individuals with fish and some kind of birds and all kinds of stuff. So it's not hard to imagine. And then and a lot of what you don't see are the bigger stuff, like monkeys and antelopes and even gorillas are sources of bushmeat. Many times it's not even they're out. These guys aren't even out poaching the animals. Many times they're just looking for dead carcasses and that they can butcher and eat. And so gorillas are often not poached, but actually scavenged. So the virus, as I said, presumably comes from bats. There have been some instances where bats have had tested positive for antibodies to Ebola, like healthy bats, and that suggests that the viruses are present and the bat has some amount of immunity to them. The structure of Ebola is unusual. Let's just say that the virus has this really weird worm-like structure, which is not typical of viruses. The arrangement of the ultrastructures is not unusual, but it's an enveloped virus, as you can see here, this little blue layer here, and the virus has typical spikes emanating from the surface, these GPs or glycoproteins as we call them. There's a matrix protein which is primarily made up of VP40, viral protein 40, these brown little ovals, and then occasionally a VP24. You see the VP24 molecules, and they seem to be located throughout and often in association with the internal region of the glycoproteins. Over here at the front end of this virus is the polymerase complex, which the virus uses to replicate. And then a variety of nucleoproteins, which protect the viral RNA in this helical type arrangement of capsid. This diagram sort of overviews what you saw in this slide here and gives you a one sentence or so description of what the particular virus structure does. So the membrane glycoprotein, for example, is for attachment and membrane fusion, no surprises. And sometimes the virus will secrete glycoproteins into the environment from the infected host cell. It's possible that this is for virus cloaking, meaning that the immune system gets confused and starts to produce antibodies, and the antibodies attack the pieces of virus that are sloughed off, allowing the full Ebola particle to escape. Almost like a squid shooting out a blob of ink and then darting off in the opposite direction. Nucleoproteins, as I showed you, are part of the helical nucleocapsid, and they can help in packaging and transcription, but they're mainly, obviously, protective of the RNA. The VP40 is for structure, as I showed you. So again, this VP40 matrix protein is for structure. And VP24 is a nucleocapsid assembly. And I'll talk about it later on in terms of helping the virus overcome the immune response. So again, VP24 is this little guy right here, these little proteins that are embedded in the matrix. The VP35 is also a nucleocapsid assembly, packaging, and RNA polymerase cofactor. And so, and it also has somewhat to do with immune system avoidance. And I'll talk a bit about that. But the VP35 then is right here, part of the polymerase cofactor. So here's the big polymerase, these gray ovals, and then VP35 associated with that. VP30 is a virus-specific transcriptional activator and is also involved in nucleocapsid formation. And lastly, the RNA polymerase. Here's a representation of the minus-stranded RNA genome. And you can see, where the different genes line up, how there's a little bit of RNA editing that happens here so that you can get fragments, the secretory glycoprotein, and then the full glycoprotein that's used in receptors, and then the various other genes, including this polymerase section there. So as far as how the virus binds, specifically its targets are monocytes, fibroblasts, and endothelial cells. So the monocytes, for example, are immune system cells, phagocytic immune system cells that move around in the tissues and blood and ingest or phagocytose particles that are considered non-self. And endothelial cells, obviously, are cells that line the vasculature, the arteries and veins. And so in this particular case, you have an Ebola with its glycoprotein recognizing its specific receptor. The virus is internalized by a process called macropinocytosis. So this will almost like a wave forming over the virus so that it comes in a vesicle. And then once inside this vesicle, the virus can actually, in this particular case, the virus glycoprotein can be activated by cathepsin. So these are proteases that we produce. And so tied up and locked up in this endosome with the virus would be cathepsin, which again we produce as a protease, and that will cleave the little glycoprotein and facilitate a really intimate membrane fusion event. And so in that process, the membrane can be opened up, and then the viral RNA can be extruded out, as you see here, the coil of membrane of RNA being extruded out through the membrane.